My name is Maddie. Today I'm going to be talking about how neurons die. Okay, so why is this an issue that we even care about? So neurons, those are the cells that make up our brain and a lot else of our body, are post-mitotic cells. So that means once they die, they're gone. You can't bring them back. So when we look at how neurons degenerate and how they accumulate damage over time, you need to know how to intervene before the cells die in order to have any kind of meaningful intervention in that process. So in particular, the kind of damage I was looking at in my project is oxidative stress. So if you look on the right, on the top, we see this balance scale, ROS and AOS. ROS is reactive oxygen species, so that's kind of oxidant. It's a kind of damaging species in the cell. And then on the flip side of that, we have antioxidants, AOX. So that's the reason you can drink a glass of red wine, right? Because the antioxidants are good for your heart, good for your brain. And when the reactive oxygen species, the oxidants and antioxidants, are balanced, we see a state of health. However, when the reactive oxygen species or these other oxidants kind of overcome the cell's antioxidant capabilities, we see damage in the cell. And as that accumulates over time, it turns into disease and neurodegeneration. So that's kind of the driving motivation behind this project. I looked at a really specific pathway of this kind of damage. So to recap what I knew going into this project, we have a few key players in the cell, right? So we have these oxidants that I talked about, those reactive oxygen species. Others include hydrogen peroxide, you might have heard of, or peroxynitrite. The one I was working with is called DTDP, and that's what it looks like. So this is a synthetic compound, but what it does is it allows us to create this kind of damage in a very controlled way. So there's a lot of ways you can study oxidative stress. You can induce stroke in a mouse or something like that, or create an animal model of disease, but I wanted this very controlled pathway to look at. So that's why I used this molecule, DTDP. And that's bad, that's why it's red. And then on the flip side of that, again, we have these antioxidants. You might have heard of, you might not have. So they include superoxide dismutase, catalase, glutathione. So those are the good guys we have fighting on our side. Now DTDP, so that was my molecule of interest, my oxidant of interest, has been shown in the literature to introduce this programmed cell death pathway in neurons. So A leads to B leads to C. And when cell death is programmed like that, it means you can take steps to intervene. So if you can block B from going to C, you should be able to rescue the cells from that kind of damage. And you see here that DTDP is up on the top. It leads to this increase in zinc, so on and so forth. At the bottom of that, you'd see cells die. Okay, so we have our oxidant. We have this set up pathway, this programmed death pathway. But wasn't clear in the literature and was a little ambiguous, and what I really wanted to focus on is the kind of environment in which this programmed pathway can evolve. So the unknown here is the role of this other kind of cell called astrocytes. Astrocytes are this kind of glial cell. They're not neurons, but they are there to support. My PI's favorite joke about astrocytes is that they're like the women of the brain. They do all the work and get none of the credit. So that was the cell I wanted to look at. And there's some evidence that they can regulate how neurons deal with this kind of oxidative stress. And this wasn't entirely clear from the papers I'd read. So what I wanted to look at, in particular, is if these astrocytes, these kind of cells, are involved in allowing neurons to enter this programmed cell death pathway following stress from this oxidant, DTDP. Okay, so how did I go about investigating this? I grew cells. So you can see I have two kind of neuronal cultures. So that's when you take cells from an animal and you grow them in a dish, and then you can manipulate them how you want. And I grew these neurons in two different environments, astrocyte rich and astrocyte poor. So my variable here is how many astrocytes are there? Will that make a difference in what's going on? So here's the two cultures. You can see that I stained for GFAP, that's glial fibrillary acidic protein. It's basically just a marker for astrocytes. So what I was doing here, I just wanted to make sure, you can see on the left, there's a lot of that red staining. Those are the astrocyte-rich cultures. And then on the right, we have those astrocyte-poor cultures. You can see there's only one really big astrocyte, but all of those other blue dots are neurons. So that's the cell that I'm really looking at. Okay. So we have our cells set up, we have our oxidant set up. Now we have to look at how to block this death. So I showed this earlier, don't get overwhelmed by it. Basically what I want to look at here is how DTDP, the first step in this programmed death pathway, is an increase in free zinc. And we wanted to look at maybe if we can block that increase in free zinc, we can block this programmed death pathway from continuing. So the tool we use to look at that is this drug called T-pen. You can see it there. And what it does, it'll chelate the free zinc. Basically, it'll just bind it up. So in our case, the heavy metal is zinc. And it prevents it from interacting with the next target in the pathway. Okay, so if this T-pen works, it'll prevent zinc from interacting with the next step. It'll block B to C. It'll block the death pathway, right? If it is a programmed death pathway. So I have my oxidant. I have my tools to look at this programmed death pathway. Now I need to try it in my cells. So the first thing I did here, if I can draw your attention to the red line, you can see that as I added DTDP, my oxidant toxin, to my cells, these are the astrocyte-rich neurons, it's toxic to the cells in a concentration-dependent manner. So as you add more and more DTDP, more cells die, or there's less survival. Now, as soon as you add T-pen, that's the green line, so that's the zinc chelator, it's binding up the zinc. You see this remarkable sustained survival in the cells. That significance is compared to DTDP alone. So this told me that programmed death is happening in these astrocyte-rich cultures. I can intervene with this pathway using T-pen, and the cells survive. This is in the astrocyte-rich cultures, right? So of course the question then is, what happens in the absence of astrocytes? That's really what I'm looking at. And what we saw, if you look at the red bar, is that DTDP induces toxicity, but when you add T-pen in addition, so that's that green bar right next to it, there's no difference in survival. So adding this potential rescuer that works in the astrocyte-rich cultures has no effect in the astrocyte-poor cultures. What does that tell me? It told me that this death pathway is uncontrolled. This programmed mediated death pathway that I laid out in the astrocyte-rich cultures isn't happening here. And what's the difference? There's fewer astrocytes. So just to lay this out again, in the astrocyte-poor cultures, we see very little survival when we add T-pen. But in the astrocyte-rich, there's a very significant increase in survival when you add this intervention. So death is either uncontrolled or controlled. Now, one more way of looking at this, these are pictures of the actual cells themselves. You can see on the left, those are the healthy astrocyte-poor neurons. But when we add DTDP, you don't see those healthy cell bodies. There's all this junk in the background. And then when you add T-pen also, there's no survival observed. You can see that the third image there looks a lot more similar to the second one than the first. Okay? So this astrocyte-poor death is uncontrolled. I wanted to confirm that this was happening with one more target of this death pathway, and that's this 12-lipoxygenase. Basically, I wanted to see if it was part of this programmed death pathway. So I saw how an inhibitor of that molecule worked in both the astrocyte-rich and the astrocyte-poor cultures. So that's the ML351. It's a specific inhibitor of this 12-LOX. And what we see is that when we add this inhibitor, we can rescue the astrocyte-rich cells. More evidence for a programmed death pathway. And now this is a new target in that pathway. That was a novel finding of this study. Now, of course, we tried it in the astrocyte-poor cultures, and we see, similar to what we saw with T-pen, this intervention has no effect on survival, right? DTDP is toxic, and adding this inhibitor does not bring up cell survival at all. So that's more evidence for an uncontrolled pathway in the astrocyte-poor cultures. So that was a lot. But to recap, what I saw in the course of my studies is that astrocytes, this historically overlooked kind of cell, can steer neurons, which are the cells which are more typically studied, into this pathway of programmed cell death, following this specific type of insult, right? Without astrocytes, the neuronal death is uncontrolled. So it's unmediated, no intervention we take has any effect on cell survival. The good news is that your brain is an astrocyte-rich environment, right? So the implication here, why does this matter? I've just been looking at cells and dishes. We have a ton of astrocytes in our brain, and this could be a new way to approach intervention, right, in neurodegeneration. We can use astrocytes and manipulate their protective abilities in order to increase survival of neurons, right? That's all I have. I can take questions. Yes.
Are there analogous systems in like outside the CNS and PN? Like
Sure, yeah. So oxidative stress affects all cells in the body. You'll see it in the heart, really any system. I just am particularly interested in the brain and the central nervous system, but you could look at similar pathways in different kinds of cells throughout the body. Mm-hmm. Yeah.
So how does DTDP trigger cell death in like the astrocyte core environment? Like how does it go just straight from like introduction of that to cell death? Like if there aren't the known intermediates, then what do we think that looks like?
Yeah, so that's the interesting question. Maybe it's like a necrotic death. Maybe the cells are just, the insult is so severe and they're not getting whatever, maybe the astrocytes are releasing some factor that allows neurons to enter this programmed pathway. So the way I conducted my experiments, I would introduce these insults and then look a day later, which is not ideal. I can see big picture what's going on, but I'm not seeing minute to minute, what's happening step by step by step. So I need to do more work to see what's going on there. But that is kind of the crux of the question. Yeah. Is that time? All right. Thank you.